Hello viewers, today's topic of discussion will be B cells. Let us know few things about these B cells. They are very interesting and how they are developed, how they are matured and how their multiplication happens. These are few very interesting things. As we all know, B cells and T cells are the cells responsible for adaptive immunity. Morphologically, these two are not very different from each other, and these cells originate from same precursors. But B cell development take place in complex microenvironments created by stromal cells of bone marrow, which are known as niches. So here the B cells are developed in bone marrow, while T cells are developed, educated in thymus in case of human beings. So B cells precursors and T cell precursors, which are hemopoietic cells, they are same, but their development happens in different places. So B cells are developed in bone marrow, and that is why they are called B cells in human being. During the development of B cells, a wide variety of B cell receptors are developed. Now, what are these B cells receptors? These are, you can say, chemical structure in B cells which are ready to receive appropriate antigen, which will be which they may come across. That means if just we suppose that a triangular shape antigen is coming to approach, the B cell receptor will be cut in such a way that that triangle can fit properly in the receptor. So it happens that 5 into 10 raised to power 13 different types of B cell receptors are there, which can receive 5 into 10 raised to power 13 different antigens. These receptors are already present in the B cell. So each mature B cell eventually expresses only a single type of B cell receptor. When B cells are developing, they have receptors, and it's a very lot of chemistry and genetics are involved in development of B cell. I'm not going into that details of this because that requires a lot of genetics and chemistry to understand. And to keep my topic short, what I want to deliver at this stage, and that is what a medical or dental student will need to study. And even a person doing post graduation in microbiology will need only this. Where I'm concentrating is that B cells, while developing in bone marrow, have lot of chemistry is there, variable chains and heavy chains, and all this will be there. And what they will develop a receptor, and each B cell will be having a single type of receptor. That is, if one B cell is having a receptor to receive a triangle, that's just example, triangular shape antigen, while another B cell will be having a receptor if an antigen is star shaped. So just the opposite of star will be there, so that as a star can fit in a puzzle, in the same way this star will be able to fit in the B cell receptor. So each B cell develops a different kind of B cell receptor. And these B cells emerging from bone marrow are guided towards, after having these receptors, these cells will be guided towards secondary lymphoid organs. What are these secondary lymphoid organs? These are spleen, lymph nodules, Pierce patches, tonsils, and mucosal tissues. So these B cells having B cell receptors will be transferred to secondary lymphoid organs that I have mentioned. When they go to secondary lymphoid places, what happens? They come across the antigen. And when they come across a particular antigen, this antigen will stimulate B cell receptor. And this B cell receptor will receive this antigen, and there will be a micro environment while these B cell receptors are receiving an antigen. In that environment, number of cytokines, they are the co-stimulatory factors for B cell. They play an important role in that environment. So the antigen that is being encountered by the B cell receptor plus the microenvironment where this B cell receptor is present. In the secondary lymphoid tissues, there the mature B cell has entered the lymphoid tissues, and further development and proliferation will occur according to the antigen it is receiving and the environment, the microenvironment of cytokines or co-stimulatory molecules which are making up. So these B cells may be differentiated into plasma antibody secretory cells. Plasmocytes will be, which can secrete different kind of immunoglobulins. I have already put a whole lecture on immunoglobulins. So those who want to know about immunoglobulins can go and visit that, can go and see that lecture on immunoglobulins. Here we will know how these immunoglobulins or plasmocytes are formed, which will further secrete immunoglobulins. Or these B cells can even differentiate into memory B cells. These innate immunity is not specific. When we talk about innate immunity, it is not specific and it is not having any memory, while adaptive immunity do have memory. So you can understand that B cell receptors, while after receiving antigen, can develop or differentiate into develop and differentiate into either plasmocytes, that is antibody secretory cells, or they can become memory B cells. So this signaling mediated by B cell will send a signal where the different biological effects can happen to a cell. That means how this cell is going to survive, what will be the survival of this cell, how tolerant it will become, whether it will go to apoptosis, whether the proliferation and differentiation in antibody producing cells will happen, or memory B cells will be formed. This whole thing will be decided when B cell receptors will receive the antigen, and then the microenvironment of cytokines will decide. Even T cells plays a very important role. So when we will talk about T cells, we will know how these antigen presenting cells and T cells also play the role in B cell proliferation and differentiation. So here today we will talk about many co-stimulatory molecules or cytokines which act on B cells and play important role in development, survival, differentiation, and proliferation, that is multiplication of B cells. Co-stimulatory molecules, one of them important one is B cell activating factor, that is known as BAF, that is a cytokine and member of TNF family and essential for maturation and survival of B cells. This is an essential activating factor. In the microenvironment of cell, this has to be there for the B cell to mature and survive. This also participates in differentiation and proliferation, that is multiplication of B cells. And this has an important role because elevated levels, if BAFF is more, then it can cause autoimmune diseases like SLE, Sjogren's syndrome, rheumatoid arthritis. So this is important to learn that BAFF, that is the factor, B cell activating factor, which is responsible for activation of B cells, if present in more quantity, can cause certain autoimmune diseases also. Another co-stimulatory molecule we should know about is APRIL, that is A proliferation inducing ligand, and CD40 and CD40, CD154. This is the two ligands which are CD is full form of CD is cluster differentiation, and these are the receptor sites which are expressed on T cells. But I told you that we will be talking about, when we will be talking about T cells, we will talk how these CD40 ligands are also important to increase the expression of cytokines. And when the cytokines are expressed, these cytokines are important for development and proliferation of B cells. Some more cytokines which do play a role in development and proliferation of B cells are interleukin 2, interleukin 4, interleukin 6, interleukin 7, interleukin 10. Interleukin is cell-to-cell talk between the leukocytes, right? Cytokines are cell-to-cell talks, and interleukins were discovered before. So these were like in a cell-to-cell stock between lymphocytes. Interleukins is stock between leukocytes. So leukocytes is leukin word is used. Later on, we came to know that cell to cell talk happens through cytokines also. So interleukins are the ones when there is talk between two leukocytes. That these 2, 4, 6, 7, and 10 play important roles. Interferons, alpha, beta, gamma also play important role in B cell maturation and differentiation and proliferation. And CCL28 is a chemokine that directs the migration of plasma cell to mucosal sites. So CCL28 is also important. I mentioned all these leukocyte interleukins as well as co-stimulatory molecules for the purpose of entrance exam, so that you can solve many type of MCQs in it. This was all about B cells. And how T cells help in B cell proliferation, that we will be discussing later on. Thank you for patient hearing.